Okay, we should start answering the questions. Okay, so first of all, Abrar Hussain asked a question on what is the difference between computational chemistry and bioinformatics. So bioinformatics is basically a field that gives you the access and the opportunity to analyze any biomolecule. That could be a protein, gene, mRNA, microRNA, any sort of stuff. Computational chemistry helps with you the analysis of chemicals computationally. For example, you want to design new algorithms for drug discovery. Computational chemistry can help you with that because you are applying computational pipelines, computational techniques, algorithms on the chemical knowledge. And this is where computational chemistry and bioinformatics diverge. Because specifically in computational chemistry, you are analyzing chemical structures that could be or could not be related to bioinformatics. For example, you just want to analyze how a phytochemical might react in an oil. So that has nothing to do with bioinformatics. Okay. So I want to design, Asad Afzal, I think asked, sir, I want to design a computational drug for cancer. What will the steps? Okay, to design a computational drug for cancer, first of all, you have to identify what cancer, what type of cancer, liver cancer, breast cancer, pancreatic cancer, what cancer. Once you have identified the type of cancer that you want to be working on, either do RNA sequencing and identify your own upregulated genes or variant using NGS variant calling. That is one solution. Or the second solution is to do literature review. Go through different papers, identify where exactly there is a lag in a particular cancer. And for example, we had a research project on colorectal cancer, and CHEK2 protein is highly upregulated in colorectal cancer, and it's also mutated protein. And this is that protein that basically is present at literally at the end of the cell cycle. And this is that protein that gives a apoptosis signal for the cell to arrest, to do a cell cycle arrest and die. But after mutation, and if it is upregulated, what happens is that the cell cycle does not arrest over time, and the cells continue to grow, and this is how the tumor grows. So we identified this gene and in colorectal cancer, and then we started developing the drug along the latest published paper now. So first of all, you have to identify which type of cancer. Are there any drug targets available where people have not worked on previously? Take that protein, start working. Okay. Another question by Amina Anjam. Sir, I have done bachelor's in biotechnology. What kind of skills are there? Okay. To start freelancing, first of all, what you need to do is you need to understand there is a huge saturation in bioinformatics. A lot of people are providing these freelancing techniques, freelancing skills on Fiverr, Upwork, all these different platforms at very basic level. For example, not a lot of people have understanding of NGS and RNA sequencing, yet they have put this information there that, okay, we can provide NGS techniques, NGS analysis. And this in turns lead to a bad quality work for foreigners. For example, if you're working with foreigners and they don't then hire Pakistani bioinformaticians. First of all, what you need to do to have good skills, and this is what I recommend before starting freelancing, by the way, understand any pipeline of NGS, variant calling or RNA. That's it. Nothing else that you need to learn right now. Just learn any of these pipelines, RNA sequencing or variant calling, whole exome, whole genome. That's it. Once you learn this technique, create a gig, put it on Fiverr, Upwork, or anywhere. Create a portfolio as well. Do your own research beforehand. Okay, I have done this analysis, for example, on breast cancer or any type of cancer, and I have this analysis, I have this document available with me. And whenever someone asks you on chat, okay, can you do this work, you can literally send that PDF to them. Okay, I can do this work because I've done it previously, please check my work. So this will give you a credibility as well, give your work credibility. So learn any single pipeline, you will be good to go. Okay, Umar Farooq has given a very meaningful suggestion that the courses that are available on different platforms, even our own platforms, we also provide training courses. What I mean to say over here is that the course name is different from the course content. It gives an illusion to the new learner, okay, I have learned Python for data science, now I'm a data scientist, and now I have good experience in Python for data science. Because I have interviewed many different people who have done this course. This is why I'm raising this issue, because I don't want others to fall into this trap. So learn something that is related to the course content as well. For example, Python for data science should teach you Python for data science. It is not Python for data science basics. That it is not, it is just giving you basics. So it is just a basic course. So try and learn proper skills. Okay, another question by Tasir Bukhari. I did my bachelor's in biotechnology and I want to switch in bioinformatics. As I said earlier, when I answered about Amina Anjum, just learn any single pipeline and just go into its depth, understand its mechanisms, and then you are good to go. You don't need to uh, go into much further depth. Another question by Ikram Mahmood, if we have basic skills in Python, Linux, R, and R scripting, how can we present these skills to get a job? Very good question, and I applaud you for that question. If you have these skills, first of all, you need to input them on your CV in a proper way. For example, Python, Linux, R. Anyone could do that. Anyone can write, okay, I know Python, I know Linux, I know R. What you need to do is you need to type R, for example, in your CV. Okay, I know R. And within subsections of R language, you need to type what exactly you did in R. For example, you did functional enrichment analysis, pathway analysis in R. You did RNA sequencing differential expression in R. Make someone read that in terms of what language you know and what you have done in that language. For example, you know Python, you are very good in Python. Just type, okay, I have developed these different pipelines using Python. And this is how you can provide insights to someone who is reading your CV that this person has learned Python and this person has employed these Python skills on these different problems. As I said earlier throughout the session, that we need to understand how to analyze problems. And if we are able to analyze problems, we can then input this information into the CV. I will give you an example. One of our employees who has been employed now, he came up with a CV and he had written variant calling analysis in his CV, and he had a proper paragraph that, okay, I took this protein, I analyzed this variants, and I did structure prediction, etc., etc. So this gave me an insight that, okay, this person has an idea how to do variant calling, how to do that, etc. So then, of course, I interviewed him properly, and it came up that he has good skills. Okay, so another question by Mia? I'm not sure how to pronounce that. So, what is the difference between BioPython and Python? Okay, Python is a language, basic programming language, and BioPython is a sub module of within Python. It is a third-party package that you can install within Python, and you can call its libraries. You want to analyze, for example, you have a genome and you want to get 1,000 sequences out of it based on the identifier. You can utilize BioPython for that. For example, if you just want to get sequence from 30th nucleotide position to 100 nucleotide position, you can do that as well using BioPython. So it does not help you in proper advanced level analysis, but basic, as I said earlier, the basic level scripts can be created using BioPython, and this is what we have been employing as well. And it is definitely something that you should learn, but not stick to it. You should definitely go towards other packages. If you are interested in Python, I suggest learning ScanPy, which is a module of Python package for analysis of single cell RNA sequencing data. So now you can analyze single cell RNA sequencing data using Python. Amna Anjum asked another question from where we can learn BioPython. BioPython, the developers of BioPython have their own book. When I was learning BioPython back in my bachelor's, I had that PDF available with me. And if I remember it, I will ask my team members to post it in the WhatsApp groups as well, so that you guys can download it. There is another way to read about BioPython, learn about BioPython has extensive documentation available on their site. You can learn from there. But if you are interested in learning from videos, we have lectures available on YouTube as well. Free of course, no one has to pay. And they are half videos. Each topic is cut into half. So you can learn from there by Python. And if you are interested in learning proper end-to-end, you might find other videos on YouTube, but if they are not available, you can enroll into our courses. Amar Farooq has given another insight. I've done courses in this. Okay. No need to be felt ashamed. Now that you have understand the core issue, you can now learn actual data science, actual Python. Faiz Ahmed Taj asked, are the current internship opportunities? Yes, there are, but we only hire people who have basic skills. And like, as I mentioned earlier, I don't give opportunities to those people who are not even interested in coming up with the biological solution. So when I do hiring, I will give you a proper, honest opinion. When I do hiring, what I see in people is they should either have practical skills or they should have theoretical skills. And if they have any one of them, I hire them. But the issue is not majority of them have either of these skills. Once I asked someone that what is cancer, and she was not able to even answer that during an interview procedure. So now you can understand why there are no opportunities. There are opportunities, but people don't know how to avail those opportunities. Yes, there are internship opportunities available. If you have good CVs, just email them to us from whichever email you received this invitation for this webinar. Just email us, and my team will let you know if we need to hire you as an intern. Okay, Asad Afzal asked, I'm now in 6th semester. What skills do you suggest something to learn to fit in the market upcoming year? Asad, I recommend that you learn genomics. Without genomics, there is no way you can compete. Anyone can compete in what is coming next. Learning everyone learns proteomics throughout their bachelor's or MS. Okay, do docking. That's it, nothing else. So you have to, everyone is being educated on this single topic. So there is a huge saturation, as I said. Everyone is working on molecular docking and computational drug discovery. So that you have to make yourself unique in some aspect. You have to learn something unique. And genomics is something where some field where no one is coming for some issues, largely due to resources, because genomics work is not easily doable on a normal laptop. So we do need a good computer to do the analysis. So you need to come up with those skills, genomics related skills. And lastly, I suggest everyone to do not neglect theoretical knowledge or the biological aspect. I will give you a basic example and we'll end this session. For example, your job is to analyze multiple myeloma, which is a type of bone marrow cancer. Your job is to analyze this data set. You can do RNA sequencing or variant calling analysis in one single day. You can be done with that. But you have to convert this analysis, this output of the pipeline into biological understanding. And to convert into biological understanding, you need to understand what exactly is multiple myeloma, what is bone marrow cancer. And then you can connect this information back to your analysis. Okay, I think so we do have some, no, that's it. Okay, so is there any other question? I'm going to wait 2 minutes and then we are going to end the session. You are welcome. I think so we should end now. Thank you so much all for joining this. Only the question and answer session was recorded, and this will inshallah be posted on our YouTube channel just for the insights of other results. We had a lot of submissions. A lot of people wanted to join, but for some reason, after it's Pakistan and electricity issues and etc. So if anyone has objection for this recording, they can let us know. Thanks so much all, and we can communicate further in the WhatsApp groups if you have any questions, but I will not be available today, and whenever I have time, I'll let you guys know. Thanks so much. Have a great day. Bye.